Hello, my name is Gifty McQuay. Today's date is September 5th, 2025, and welcome to my cytology presentation. What is cytology? It is the study of cells that focuses on and examines the function, structure, and behavior of a cell. Here are some facts about cytology. Let's break down the structure of a cell. First, we have the cell membrane. The cell membrane acts as a protective barrier that is selective of what enters and exits the cell walls. It also allows communication between cells. Here are some facts about the cell membrane. Within the cell membrane, you have the membrane proteins, which plays a big role in communication, breaking down nutrients, transporting of molecules, deciding who gets passage, and numerous of other functions. Here are a list of functions that consist of the cell membrane proteins. The membrane transport is the gateway to the membrane. There are two main membrane transports, active versus passive. Please note that the membrane transports does not include ATP, but it still has other ways of transporting, such as filtration, simple diffusion, and osmosis. As you can see, here's other examples of passive and active transports. Next, we have the cytoplasm. This is a jelly-like water-based substance that's inside of a cell. It contains all of the organelles and holds them in place. Here's a list of everything that's within the cytoplasm. We will be walking through them later on in this presentation. Next, we have the cytosol. This is the water-based fluid inside of a cytoplasm. It's the fluid that organelles floats within. It's also known as the intracellular fluid, or ICF. The main control center of the cell is called the nucleus. It's the blueprint for our bodies and oversees all cellular activities. It is surrounded by a bilayer membrane that's called the nuclear envelope. The nucleus contains DNA and the nucleolus. What is the nucleolus? Well, it is the largest spherical structure centered in the nucleus. Its primary function is to promote. Next, we have here the endoplasmic reticulum, also known as ER. It consists of a network of channels within a cell functioning as a transport system for lipids and proteins. The system of extensive channels are known as the cisternine. There's two types of ER, smooth ER and rough ER. Smooth ER is a channel within the membrane that lacks ribosomes. Its primary function is to detoxify the cytoplasm. Rough ER consists of folds of tissue and channels continuously with the nuclear envelope. It is named rough ER because it contains ribosomes. Ribosomes are cellular machinery that produces peptides and proteins. They also have the ability to read DNA code and interpret RNA messengers. Next, we have the Golgi apparatus, also known as Golgi complex. It's a stack of small flat sacs formed by membrane called cisternine inside the cytoplasm. It's a central sorting station located near the ER. It processes and packages the proteins and lipids it receives from the ER. Here are the process of how the Golgi complex functions. Next, we have here the lysosome and the peroxisome. They both have digestive enzymes that break down various particles and organelles, except peroxisomes are produced by the ER instead of the Golgi complex like the lysosomes. Another difference between the two is that peroxides produce hydrogen peroxide where lysosomes don't. Now we have the mitochondria. It is known as the powerhouse of the cell. It is the primary source of energy for a cell. Here are some key factors about the mitochondria. Now, with the last but not least organelle we have here is the centrioles. It is normally a pair of cylindrical organelles found near the nuclear envelope. Each centriole contains numerous microtubules. Microtubules are proteins responsible for cell division. Here are other facts about these centrioles. And now, that brings us to the end. Thank you so much for watching this video. I hope you learned a lot. If you have any other questions, please feel free to contact me. Thank you again. Goodbye.